About 2 years ago in Santa Fe, New Mexico, there was a conference, mostly amongst developmental psychologists and some clinical psychologists, on this problem of boys at risk. It was the first time, actually, that the conference like that came together. And since then, there have been 2 more conferences on it. Since then, the Infant Mental Health Journal, which is the major technical journal in that field, published an edition of 10 articles on this problem of boys at risk. Boys at risk in the United States, including middle-class boys, poor, upper class, African American, Indian American, Hispanic, et cetera, et cetera. So I want to share some thoughts about this problem. And again, this was originally in a keynote address to that. The idea that the growth and the maturation of males is affected more adversely by early environmental stress than females has a long history in the biological, medical, and anthropological literatures. Actually, the first studies to show that boys are more at risk to girls, to a major stressor, was in Hiroshima after the atomic bomb, finding that boys were suffering more than girls from that stress. The more modern concept of boys at risk clearly suggests the importance of gender in order to understand both the psychology and the biology of human beings, especially early developing human beings. The term at risk implicitly implies a vulnerability not only in the early development, but also at some later stage of the lifespan, and explicitly connotes that a certain group of high-risk male children will, as they enter adolescence, be highly susceptible to a number of psychiatric disorders whose symptomatology first appears in that period of human development. One also suggests that a vulnerability of specifically developing boys and not girls to certain psychiatric disorders, and thus psychological difficulties that are more associated with the male gender versus the female gender. In 2003, I was part of a book, Hardwired to Connect, a report to the nation from the Commission on Children at Risk, along with other pediatricians and psychiatrists, etc., in which we concluded in recent decades, many adults have tended to withdraw from the task of assigning pro-social meaning to gender, especially in the case of boys. But neglecting the gendered needs can be dangerous. As the medical world has discovered, the risk of not attending to real differences that exist between males and females can have dangerous consequences. For example, in medicine, we're now looking at differences of males and females to heart attacks and strokes, et cetera, et cetera. In 2005, in the journal Science, Carolyn Holden offered an article, Sex and the Suffering Brain. And she said it's easy to start a fight about whether there are gender differences when it comes to mental skills, intelligence, IQ, verbal skills, etc. But there's little debate that patterns of mental illness and disorders vary between the sexes. Women, for example, are more likely to get depressed. Men are more severely afflicted by schizophrenia. Males exhibit more antisocial behavior. Most alcoholics and drug addicts are males. Even suicide has a gender bias. Females make more attempts, males are more successful. So a number of fields have now been studying this. This you see, the Journal of Neuroendocrinology, which essentially is the science of hormones. There are differences in the diagnostic rates between genders of such apparently disparate conditions as autism spectrum disorders, which is male to female 80 to 20, 4 times as many. Early onset schizophrenia, 80 to 20. ADHD, more males, 58 to 42. Major depressive disorders, now it switches around to the other way, 33, 67. And anorexia, 7 to 93, which is essentially a female disorder. In neuroimage, Schneider said that conduct disorder, characterized by a range of behaviors such as physical aggression, stealing and lying, and destruction of property, is most frequent in adolescent males. And actually, where this is going, the next conference in the United States is going to be on violence. Why are males more prone to violence? Holden said that men are more prone to expressing unhappiness through and externalizing pathways. Those of you in psychology familiar with the fact that males go into externalizing psychopathology, females go into internalizing psychopathologies. But males externalizing drinking, drug use, violence, whereas women are more likely to internalize, leading to depression, anorexia. More males than females have schizophrenia, and they have earlier, more severe symptoms. These deficits all begin prenatally during the period of sexual differentiation of the brain. Now, there's some changes here because it used to be thought that gender differences really came online in the psychology literature in kind of the second year. And we're now seeing that there are differences in male brains and females' brains in utero, and I'll give you this data. This clearly implies the importance of early male brain development and the origins of externalizing psychopathologies. So Carolyn Zahn-Waxler, developmental psychologist, child psychologist, authored an article, Disorders of Childhood and Adolescence, Gender and Psychopathology. And she pointed out even toddlers and preschool children, so she's talking here about toddlers, that would be the end of the first year through the second year, can have serious externalizing problems, and sex differences are already present at this time. This suggests an even earlier onset that may need to be incorporated into developmental models. The delayed physical maturation and language development of boys versus girls, along with emotion regulation problems, may place young boys at greater risk than girls. She went on to say that the origins of male and female preponderant problems are likely to be rooted, in part, in biological, physical, cognitive, and particularly social emotional differences in boys and girls that can precede the expression of clinical problems. Study of sex differences provides a means to identify the complex etiologies for different forms of emotional behavioral problems. So it's now accepted that gender differences in developmental disabilities and psychiatric disorders originate early in social emotional development. And recently, male-related developmental disorders have significantly increased. It's the work of the Canadian Bruce Lanphier in the Annual Review of Public Health 2015, pointing out that the prevalence of autism has increased dramatically. The Centers for Disease Control and Prevention reported a 123% increase from 2002 to 2010 in autism spectrum disorders amongst 8-year-old children, the age of peak prevalence. 10 years ago, the prevalence of autism spectrum disorders was 1 in 1,000. Today, it's 1 in 61. ADHD, the most common brain disorder of childhood, affects about 1 in 10 children in the United States. Boys are 2.5 times more likely criteria for ADHD than girls. These data indicate that we are in the midst of, and listen to this phrase, an epidemic of brain-based disorders. So the question is, why are boys at risk for certain later developing certain psychiatric disorders? I'm going to suggest that developmental neurobiology and developmental neuroendocrinology offer deeper insights into this question. So in the following, I want to use the perspective of regulation theory, my theory, what early developmental mechanisms are involved in the increased vulnerability of males to certain developmental disabilities and psychiatric disorders, and what epigenetic mechanisms can account for the recent widespread increase of these disorders in the US and beyond. In 1994, I wrote that the early emergence of normal early gender differences, normal differences, reflects differences in rates of neurobiological maturation of the female and the male brain. In 1969, the English neurologist Taylor offered an article in the journal Lancet, Differential Rates of Maturation Between Sexes and Between Hemispheres. We were just talking about hemispheres. These hemispheres, the rate of maturation of the hemispheres differs. But on the matter of cerebral maturation, the process proceeds more rapidly in girls than in boys, a fact that is supported by general evidence of their physical and behavioral development. The neurobiological mechanism of the slower maturation of the developing male to female brain is expressed immediately after birth. In 1999, Ed Tronick, probably one of the 2 or 3 most important developmental psychologists in the United States, wrote an article on gender differences in emotional expressivity and self-regulation during early infancy. And they concluded gender differences in social responsiveness have been documented within hours of delivery, meaning social responsiveness to the mother's face, to the mother's body. For instance, male newborns are less responsive to auditory and social stimuli and less able to maintain eye contact than female newborns. Social auditory stimuli would be the mother's voice and the mother's face. So male newborns also experience greater difficulties in maintaining affect regulation than female newborns. On the matter of newborns, in the first week of life, male infants smile less than female infants and display more irritability, crying, facial grimacing, and lability of emotional states. These are normal differences between boys, girls, right out. Male neonates also show a more rapid buildup of arousal. They go to high arousal faster and a quicker peak of excitement. Male newborns engage in less self-comforting, a behavior that functions to regulate periods of arousal, tension, excitement, or distress. So I'm going to suggest what we're looking at here is neurobiologically behind these behaviors is that there is a slower prenatal and postnatal maturation of the boy's autonomic nervous system and central nervous systems. Tronick then went on to look at what happens at 6 months. Does this trend continue? And his lab shows that there are greater differences in 6-month-old infants. Boys were more emotionally reactive than were girls during face-to-face social interactions with their mothers. Boys were more likely than were girls to show facial expressions and anger, to fuss and to cry, to want to be picked up, and to attempt to get away or distance themselves from their mothers by arching their backs and turning or twisting in their infant seats. Their interpretation of this behavior is that these gender differences focused on differences in boys' and girls' self-regulation of affect. Boys have greater emotional reactivity, suggesting that boys have greater difficulty self-regulating their affective states, and therefore they need to rely more on maternal regulatory input than do girls. Because they can't auto-regulate as fast, They need the mother there to be able to downregulate their states. Boys are more demanding social partners, have more difficult times regulating their affective states, and may need more of their mother's support to help them regulate affect. This increased demandingness would affect the infant's boys' interactive partners, who the mothers clearly see the differences here between the two sexes. So as Lab concluded, girls, on the other hand, seem less vulnerable to relational interactive stresses. These gender differences in emotional expressivity and self-regulation differentially affected the regulatory demands of mothers and sons and mothers and daughters. The mothers had to work harder with their sons than with their daughters. Over infancy, boys show functional expressions of prolonged structural cerebral immaturity in affective processing and stress regulation. Again, I want to point out, none of these are abnormal behaviors. If you looked at these behaviors, you would say that looks like an early insecure development. Not the case. It's just slower maturation. Indeed, the curve of development of the frontal cortex, the caudate, which has a heavy dopamine input, and the temporal lobes in girls is considerably faster than in boys. That the frontal cortex will mature faster in girls than in boys. This male's slower development in processing and expressing affect and his social responsiveness is related to slower maturation of the male brain. You see McClure documenting significant female advantages for facial expression processing in infants, in children, and in adolescents. That girls, infant girls, can process faces and the subtleties of faces, read their mother's faces faster. This facial expressing processing may not be fully mature at birth and continue to develop through the first 6 months. But again, through the first 6 months, she's moving quicker than he is. These regions that respond to faces may develop more rapidly and earlier in females than in males. We talked about the ability to process these faces. Early developments in facial emotional processing depend heavily on maturational changes in a neurobiological system specialized for recognition of complex patterns such as faces and selected facial features, including expressions. And studies have implicated a variety of structures that participate in this ability to process facial expressions. And the bulk of the research, however, has focused on the temporal cortex and on the amygdala, as well as a variety of structures seated in the right hemisphere. And as I said this morning, the right hemisphere is dominant for processing facial expressions. So what we're talking about here are the structure-function relationships of the early developing right hemisphere in infant boys and in infant girls. So the proposal here, and this turned out to be about a 75-page paper, is that the right hemisphere, which is dominant for processing and regulating emotion, and for processing faces, and for regulating stress, develops more slowly in male than female infants. It's the same sequence of posterior to anterior development, but different maturational rates. Taylor said that cerebral maturation would be more rapid in girls as our physical and psychological development, so that boys would be at risk for a longer time. Why are they at risk? Because they have a more immature brain over a long period of time. And this more protracted period of vulnerability occurs in the boy's slower maturing right hemisphere. The differential effect of the same attachment stressor would be different in boys and girls. Abuse and neglect would be different in boys and girls because at that same time the boy's brain is more immature. The infant boys prolong their maturity in processing and regulating stressful negative affects has been shown in a number of studies. This is Kunzler, I think in Germany. Clinical trials in humans and experimental studies in animal models have revealed that the vulnerability towards stress exposure differs between males and females. Although both brains respond to stress with a negative emotional state, authors note that male and female brains display differential sensitivity towards early life stresses. Now, these negative feelings that are the response to the stressors involve the activation of multiple neurotransmitter and hormonal systems, including the stress mechanisms that are heavily influenced by sex hormones. There you see perinatal exposure, which means right after birth, to a chemical such as testosterone, the male hormone, or to an environmental cue such as nurturance or maternal depression, can create a lasting change in mental health, either risk or resilience. But it's known that the developing brain is exquisitely sensitive to the effects of circulating steroid hormones during the so-called sensitive periods of perinatal development, such as corticotropin releasing factor and corticosteroids, which are the major stress hormones in the brain. Both androgens in males and estrogens in females, and incidentally in males, androgens, it's 10 to 1 in males, androgens to estrogens, and in females, it's estrogens 10 to 1 the other way around in females. They're both produced by the bodies of both sexes, although in different amounts. The action of sex steroids is described by two mechanisms. First are what is called organizational or programming effects, and this refers to the ability of these gonadal steroids, androgens and estrogens, to permanently sculpt the nervous system during critical periods of development, thereby giving rise to male and female physiology and behavior in adulthood. In other words, the sculpting of the brain is influenced, the synaptic connectivity, the wiring of the brain is influenced by androgens and estrogens. And this is what shapes a female brain versus a male brain. On the other hand, there are activational effects of these gonadal steroids, mediated through the acute and transitory effects on the fully developed nervous system, especially in social contexts. So, for example, if you give steroids, androgens to adolescent males, you see increases in aggression. If you give androgens to either male or females, you see increases in sexual arousal. But I'm now talking about early in development, whereby these hormones are going to influence the synaptic connectivity of the brain. Melissa Hines, Oxford. These androgens and estrogens permanently sculpt the nervous system during critical periods of development. Testosterone is significantly higher in males than in females during two specific periods of early human development. There's a prenatal surge at 12 to 18 weeks in utero of testosterone in males and not in females. And there's also a neonatal surge during the first months after birth, with the largest sex differences occurring in the first through the third postnatal month, a period that is also known as mini-puberty. These, therefore, are the times when testosterone is likely to influence human gender development. These endocrine hormones, the androgen testosterone and the estrogen estradiol, are products of the early developing hypothalamic pituitary gonadal axis. But they also program the development of the hypothalamic pituitary adrenal axis, that produces corticosteroids, generating gender differences in stress regulation, because the stress regulation is through the corticosteroids, but they're being influenced by the male and the female hormones, so you're going to get differences in regulation of in males and females. We now know that the interactions with the stress-regulating corticosteroids and gonadal steroids begins prenatally. Sexual dimorphisms of behavior derive from sexual dimorphisms of the brain and body that begin in utero. What I mean by sexual dimorphisms is that certain parts of the brain are more feminized, other parts are more masculinized, but again, this is beginning in utero. In fact, we're now talking about the masculinization of the HPA axis, at least with respect to acute stress, being affected by testosterone and androgen receptors before birth. Indeed, during human fetal brain development, the male fetus brain develops more slowly than the female fetal brain. And right out at birth, in the newborn, the CNS, the central nervous system of newborn full-term boys, is less mature than girls. That's why you get those tronic effects. The male's slowly maturing HPA axis is more susceptible than the female's to both external environmental stresses, such as separation or depression or abuse, et cetera, as well as it's more susceptible to internal hormonal alteration that accompany these social stresses. Indeed, very recent studies now show that the fetal and postnatal testosterone influences the development of the HPA axis. What I mean by that is fight-flight responses, okay? And what we're looking at here is that high maternal cortisol in late pregnancy is associated with more difficult behavior at weeks 1 and 7, whereby infants now display more crying, fussing, negative facial expressions. In other words, we now know cortisol crosses the placenta, so if the mother is in an emotionally stressed situation, it will cross the placenta, and whatever is evolving at that point in time will be affected by the cortisol, and what's evolving at that point in time is the amygdala. But what we're looking about here is difficult temperament. The continuity of the brain development of the HPA and the prenatal and postnatal has now been shown. The long-held idea that fixed inborn temperament represents genetic factors that are first expressed at birth is incorrect. We used to think what is genetic is before birth and what is learned is after birth. We now know that's not the case, that there is learning in the fetus and that genetics are exposed postnatally also. So this matter of temperament. In fact, we're now thinking that temperament, the basic temperament of the child, of the infant, is a result of an epigenetic mechanisms that has evolved prenatally and continues to be epigenetically shaped or misshaped by the postnatal social environment. In other words, the relationship with the mother is shaping the genome of the baby. And studies on prenatal predictors of infant temperament, Werner and our colleagues conclude what has been termed constitutional about temperament is emerging as identifiable in the prenatal period and may in part be the consequences of environmental influences before birth. Actually, we're now looking at the prenatal origins of certain diseases, cardiovascular disease, diabetes, etc. But again here, these are setting up before birth. And actually, personality organization is influenced by prenatal factors too. At 3 to 4 months, male infants who have higher levels of postnatal testosterone show higher levels of negative affectivity, as reflected in their greater frustration tendencies, fussing, crying, failing to achieve a goal when placed in a confining position. And this is seen in the second year. So we're now thinking that fear reactivity in males is a joint activity of cortisol and testosterone together. In general, ovarian steroids such as estrogen have been found to increase HPA activity and enhance the HPA axis response to psychological stress. On the other hand, testosterone attenuates HPA activities. In fact, in adult males, the magnitude of the HPA corticosteroid release to stress is negatively related to testosterone, which means that if in males are in very high states of testosterone, let's say adolescent males, let's say into aggression, etc., the HPA axis is dampened down and they have less access to their own physiology and their own body. These normal sex differences, again, testosterone inhibits the stress axis. Testosterone can block corticosteroid release. Testosterone-driven aggression or sexual experience in males can transiently decrease stress reactions. So the more angry, the lower the experienced stress. These gender-related psychopathogenetic effects of prenatal and postnatal social stressors alter normal gender differences. I've been speaking about normal differences. Now I want to go to speak about gender differences in psychopathology. In 2001, I suggested that extreme social stressor, what I call relational trauma, which would include abuse and neglect, induces dysfunction in the wiring of the right brain. This results in insecure attachments, especially the most problematic of insecure attachments associated with abuse and neglect, disorganized, disoriented attachment, which is highly related to a predisposition to certain psychiatric and personality disorders. The update here is that male and female brains display differential sensitivity to early life stress of relational trauma, to abuse and neglect. There's now widespread agreement that both prenatal and postnatal maternal-infant factors program the HPA axis, and that severe early life stress permanently impairs the stress-regulating system over later stages of the lifespan, thereby jeopardizing future mental and physical health of the individual. Incidentally, if you're not familiar with the work of Stephen Porges, anybody heard of this name, Stephen Porges? I started looking at Porges. Porges is the world's expert on the autonomic nervous system. So if you want to talk about bodily-based problems, etc., it's Porges. Infants with disorganized, disoriented, insecure attachments, with difficult temperament, associated with abuse and neglect, also associated with maternal alcohol abuse, exhibit the highest cortisol levels than all other attachment classifications. So in 2003, I suggested that due to the delayed rates of cerebral maturation in male infants, there are gender differences must be considered in developmental traumatology. Earlier, the work of Cicchetti, major researcher at the University of Minnesota, documented that disorganized attachments, the most severe form of early attachment disorders, is more frequent in boys in low-income, maltreated infants. 10 years later, Carl Lyons-Ruth at Harvard found that infant boys displayed more disorganized attachment behaviors and concluded that gender plays a role in the manifestations of disorganized behaviors in high-risk samples of boys. In studies of disorganized attachment in 4-month-old infants at Columbia University in New York, Beatrice Beebe reports male infants were overrepresented in future disorganized infants. Moreover, male infants are more emotionally reactive than female, so that they may be more vulnerable to a disorganized form of insecurity. So the message here is that the most disorganized forms of attachment, males over females. See, gendered intensification could result from earlier vulnerabilities such as insecure attachments. So early life attachment trauma in critical periods of right brain development will generate maturational failures in the limbic system, the emotion processing limbic system, and in the HPA axis. Here you see epigenetic changes in the nervous system are emerging as a critical component of enduring effects induced by early life experience, hormonal exposure, trauma and injury, or learning and memory. The context may be variables in the internal environment, such as the hormones, or the external environment, but such steroid hormones or endocrine disrupting chemicals, steroid hormones or endocrine disrupting chemicals. Alternatively, the context may be experienced as profound as child abuse or mild as context-dependent learning. So if you altered, not only if you expose the child to relational trauma, but if these internal hormone levels of testosterone were altered and inhibited, this could also lead to very problematic situations. Now, as a matter of fact, here you see in the Journal of Neurological Sciences, endocrine disruptors as a threat to neurological function. They describe the potential coupling of endocrine disruption and brain function because of the vital roles played by hormones in brain development and brain function. In other words, these hormones are critical, but if something should alter their levels, this could have a strong negative impact on brain development. Gonadal hormones, estrogens, and androgens, as I said, mold the sexual differentiation of the brain, and they also mold the HPA axis, they mold the limbic system. So these chemicals, which are known as endocrine disruptors, act as neurotoxic environmental chemicals in the environment, act as androgen or androgen disruptors. For example, herbicides, fungicides, insecticides, plasticizers contain these anti-androgenic elements. And it's now thought that human exposure is universal. Sex differences, by taking root before birth, are planted by the actions of hormones. These actions can be modified during fetal life by exposure of the mother to environmental chemicals while she's pregnant that mimic, inhibit, or otherwise distort the important role that these normal hormones have in enhancing the synaptic connectivity of the brain. Morphological differences between male and female brains translate into behavioral differences. These environmental endocrine disruptors transform these differences in various ways, and this applies to the significant alterations in the prenatal and in the postnatal testosterone surges that imprint emotion processing limbic circuits of the male brain. You remember I said that there was a surge in testosterone prenatally and then one right after birth, etc. And these endocrine disruptors would interfere with those surges and therefore would interfere with the imprinting of the brain of the male and the female brain. But I'm interested here mostly in female brains. Incidentally, I'm focusing here on essentially males and externalizing psychopathologies. another paper could be written about females and psychopathologies also. Because as a matter of fact, depression, as you're well aware, is now increasing significantly in female adolescents. So again, that would be the estrogen situation. I'm just looking at the androgen. The reason why the androgens came up first was because there's a lot of concern about male violence, about why young boys, why males are committing so many violent acts. The developing fetus of the infant is much more sensitive to endocrine disrupting compounds than the adults. Moreover, these effects early in life may not be manifested until adulthood. Extremely low dose exposures may exert significant effects on a developing organism, particularly if the exposure occurs during critical developmental periods. Exposure to these ubiquitous chemicals occurs in critical developmental times, but really, and most importantly, during maternal fetal transfer, where these endocrine disruptors are transferred across the placenta, but also in perinatal periods. We now have evidence that these endocrine disruptors are in mother's milk, and therefore they would be transferred to the baby in breastfeeding. So we're looking at this kind of a situation here. I'll come back to this later this evening here, but you see the connections here between the mother's brain and the fetus, etc. You're also looking at the hormonal connections between the two. Incidentally, look at over here, it's the orbital frontal cortex that the mother is using these abilities when she's reading her own baby states. So in 2014, in Lancet Neurology, Grangita and Landrigan concluded that neurodevelopmental disabilities, including autism, ADHD, dyslexia, and other cognitive impairments, affects millions of children worldwide, and some diagnoses seem to be increasing in frequency. Industrial chemicals that injure the developing brain are among the known causes for this rise in prevalence. These endocrine disrupting chemicals that interfere with normal testosterone functions that wire the brain during early critical periods represent a major etiological factor of boys at risk, and they contribute to male-related psychiatric disorders. So I want to talk here about vulnerable right brain functions in prenatal and postnatal critical periods. This overarching model of the neurobiological mechanisms that underlie why and when boys are at risk for later gender-related psychopathologies must address the fundamental question of specifically which brain circuits are shaped or misshaped by fetal and postnatal stressors, and how these systems show later deficits in social and emotional functions in adolescence and adulthood. This was an editorial in the Journal of Child Psychology and Psychiatry in 2011, Leckman and March, Developmental Neuroscience Comes of Age. The field now declares the enduring impact of early maternal care and the role of epigenetic modifications of the genome during critical periods in early brain development and health and disease is likely to be one of the most important discoveries in all of science that have major implications for our field. Brain development is not just genetically encoded. It needs these epigenetic social experiences. It's not one or the other. We're now using the term gene-environment interactions. We're looking at Mother Nature and Mother Nurture combining to shape human nature and also human gender. These epigenetic influences of maternal-infant attachment emotional communications would also be on right brain circuits, since the right brain circuits are going to be setting up prenatally, right? and then postnatally. So these circuits are going to be particularly vulnerable. I suggested in 1994 that during critical periods, steroids influenced the rates of development of the components of neural circuits. The action of gonadal steroids act as an essential link between nature and nurture. That is, by which psychoneuroendocrinological changes during critical periods can initiate permanent effects at the genomic level. The final developmental outcome of early endocrine gene interactions is expressed in the imprinting of the evolving brain circuitry. For example, the brain circuitry between the orbitofrontal and the amygdala is the key stress-regulating circuits. These things are going to alter that connectivity, and therefore you're going to have affect regulation problems. Developing emotion processing limbic circuits really are what are problematic here. Indeed, a sexual dimorphism, a differences between male and female brains that is dependent on sex hormones has been, during a postnatal critical period, a brain differentiation has been found in the medial and central amygdala and the postnatally developing orbitofrontal cortex, that there is a masculinized orbitofrontal and a feminized orbitofrontal, masculinized amygdala, feminized amygdala. And I suggested that the orbitofrontal differentiation and growth will stabilize earlier in females than in males, and that orbitofrontal cortex functionally matures according to different timetables in females and males. And again, if it takes longer in males, there's longer periods of vulnerability there. This induces an enduring masculinization and feminization of the orbitofrontal cortex, the stress-regulating cortex, over later stages of life. You remember blue amygdala, yellow cingulate, red orbitofrontal. In Holland, sex differences in orbitofrontal cortex activity may be due to the organizational effects of gonadal hormones early in life. The behavioral and neurobiological differences between men and women are an expression of more general sex differences in the regulation of emotion. Early life levels of testosterone tune the orbitofrontal to more male-like or more female-like function. So I've suggested that early life emotion-regulating attachment experiences shape the right orbitofrontal cortex, the apex of the limbic system, the control system of attachment, the sense of masculinity and femininity. In 2001, I offered an article, Effects of a Secure Attachment on Right Brain Development, Affect Regulation, and Infant Mental Health. Over the course of the first year of life, the mother's right brain rapidly receives and regulates emotional responses to her infant's emotional communications, as we've discussed. The mother's right orbitofrontal reads and regulates the infant's amygdala-driven affective states. These limbic circuits in the early developing right hemisphere are imprinted in mother-infant shaped in right brain to right brain emotional communications. I've shown this. So I've suggested that attachment transactions influence the early life programming of hemispheric lateralization that in turn generates the dominance of the right hemisphere in the first year of life. In substantiation of that idea, at the beginning of the first year, Michael Meany and his colleagues in Canada conclude in early life, the right cerebral hemisphere would be able to process emotion, citing my work. These neural substrates function as hubs in the right hemisphere for emotion processes and mother and child interaction, as I showed earlier this morning. Tronick's research on infants now in the middle of the first year, that under relational stress, 6-month-old infants use left-sided gestures generated by their right hemisphere. In other words, they'll self-comfort themselves. These data are consistent with Schore's hypothesis of hemispheric right-sided activation of emotions and their regulations during infant-mother interactions. And then this morning I discussed Minagawa and Kawai at the end of the first year. Our results are in agreement with Schore right hemisphere attachment. Subsequent to the child's attachment to the mother in the first year, forms another to the father in the second year. Though the mother's soothing is essential to the child's attachment security, the father's arousing play is thought to be critical for the child's competent exploration of the physical world. The father imprints the later developing left hemisphere. So in the second year, the second year, when the left hemisphere comes online, the father will imprint that, just as the mother imprinted the early right, and that will be imprinted, as I'll show later, in things like rough and tumble play with boys, etc. But it turns out that the father is later critically involved in the male and the female toddler's aggression regulation, but the mother is earlier involved in the first year in fear regulation. And we now have a number of studies showing that paternal care also affects synapse formations in the developing brain, but in different parts than the mother. And again, that's in the second. Now, on the other hand, this attachment operates for better or worse. And so relational trauma now induces dysfunction in the development of the right brain. Insecure attachment, especially disorganized, disoriented, abuse, neglect, is clearly associated with a later predisposition to psychiatric and personality disorders, for example, borderline personality disorders. Childhood adversity often appears to operate similarly to both sexes, but the infant male and female brains display differential sensitivity to early life stress and to relational trauma. Indeed, recent studies are now showing that infant male and female brains show differential sensitivity to early life stress, and that specifically this will impact the maturation of the prefrontal and the limbic system. Here, traumatic emotional experience during early life exerts a strong impact later in life. Axis 1 personality disorders and psychiatric disorders showing up late in adolescence. And they show that repeated early separation in the first year results in hyperactive behavior and changes in prefrontal limbic pathways, regions that are dysfunctional in a variety of mental disorders. So the long-term effects of this are that impairments in behavioral flexibility, the individual loses this kind of flexibility, impairments in emotion processing and different aspects of executive control, which are mediated by the orbital frontal and the medial frontal cortex. Now, there are implications for this in the early assessment of boys at risk. Developmental interpersonal neurobiology now indicates that we should move from higher verbal cortical executive functions to lower subcortical limbic autonomic circuits that are imprinted in gender socialization in infancy and toddlerhood. For example, there are clear studies showing that in disorganized attachment, the right amygdala is problematic, is miswiring. Also showing structural deficits in the right amygdala in autism spectrum disorders at 6 months in infancy. So we have to start moving away from these higher level executive functions to these more subcortical areas. The subcortical areas of the brain double in size in the first year of life. This evidence-based regulation theory assessment therefore calls for looking at right brain visual, facial, auditory, prosodic, and tactile gestural communications in which the primary caregiver interactively regulates the states of emotional arousal. These assessments in right brain critical periods, they should be there in critical periods, especially from 0 to 18 months. Both sexes move through similar patterns of a caudal to rostral, subcortical to cortical development, but there is slower maturation here of the developing male versus the female brain. Means that there's increased prenatal and postnatal exposure and vulnerability to stressful physical and social environments during the formation of these implicit right brain affect regulating circuits. The process of attachment, imprinting of these evolving right lateralized limbic system is universal. However, the timing and the length of the stages varies in males and females. This differential maturational rates in brain growth spurt from the last trimester of pregnancy to the second year suggests separate norms for each gender. These norms must reflect normal gender differences in the slow maturing male versus the fast maturing female, but the risks would be at the extremes of the normal ranges. And you see I've written 2 articles on how to make differential diagnosis between attachment and autistic spectrum disorders in the first year. So I want to end this up with returning to the earlier question of boys at risk. What early developmental mechanisms are involved in the increased vulnerability of males to certain psychiatric disorders? And what epigenetic mechanisms, which are influenced by the social environment, can account for the recent widespread increase in these disorders? And now I'm going to speak specifically with data from US culture, although some of this also comes from Canada. In 2012, I documented from the child psychiatry literature in the United States a crisis in male and female children's mental health in the US. 1 in 5 children has a diagnosable psychiatric disorder. 1 in 10 suffers from a mental illness severe enough to impair everyday living. I'm going to suggest that one of these stresses is early child care. Again, what we're looking at here is entry into daycare at 3 months. Holden said, "The culture helps shape how the 2 sexes express mental problems." Over 20 years ago, in the last chapter of my first book, Affect Regulation and the Origin of Self, I overviewed disturbing research data on documented increases of insecure and especially avoidant attachments and aggression associated with early day care. From a neurobiological perspective that focuses on not short-term cognitive but long-term enduring social emotional effects, these findings were alarming. 13 years later, in 2007, this is the work of Belsky. Evidence indicates that care initiated early in life and experienced for many hours, especially in child care centers, is associated with somewhat elevated levels of externalizing behavior problems, aggression, and disobedience, and that these effects are not simply a function of low-quality care. In a later study, this group found that the effects of early child care extend into late childhood and adolescence. In 2015 in the Netherlands, Albers showed that center care at 3 months, and these were professional women dropping off their babies at 3 months, in usually what are considered to be higher quality child care, may be even more challenging for children high in negative emotionality. I want to suggest that males are more vulnerable to the stresses of early child care, which usually begins at 6 weeks. the time of the perinatal testosterone surge, and a critical period of the amygdala HPA connectivity. Recall, exposing newborn males to separation stresses can be regarded as a severe stressor. Sumi's work on the later health effects of early child adversity shows males are much more affected by early deprivation than females. So the question, as we're continuing here, would be, would these same findings be there at entry at 12 months? And would males be more susceptible at 12 months? Here you see Krockenberg in view of an extensive vulnerability to stress when gender differences in children's reactions to child care are identified. It is boys who show more negative behaviors. This is Megan Gunner, major American psychologist. U.S. child care elevations of cortisol in boys are associated with angry, aggressive behaviors, while large rises of cortisol in girls with anxious, vigilant behaviors. Boys may experience more stress at child care than girls. These gender differences reflect early expressions of, on the one hand, externalizing and internalizing psychopathologies. In a recent volume I co-edited, Evolution, Early Experience in Human Development, I propose that the current large increase in child psychopathology in the U.S. is directly related to the entry of many of our culture's infants into substandard early daycare, 6 weeks or less, due to the lack of national parental leave policy. And in the United States, what I'm suggesting is that we need to legislate strategies operating in other industrialized nations. For the United States to even get to 6 months would be a huge boost, and paternal leave at 2 months. In addition, we must seriously address the problem of training and upgrading the quality of child care workers, and that goes in all countries. Usually these professions are undertrained. This is Albers in the Netherlands on entrance early in the first year. On the one hand, high quality of maternal caregiving behavior helps infants to regulate stress in challenging situations and in the long run to develop self-regulatory capacities. So this is optimal with the mother and the infant. On the other hand, young infants accustomed to receiving high quality care at home may be less prepared to deal with the challenges without such maternal aid, especially if the quality of the care provided by the caregivers in the center is relatively low. This is the study here in Norway, done in 2016. Kleti, is that how you pronounce that? Together and Alone, a study of interactions between toddlers and child care providers during mealtime in Norwegian child care centers. In most industrialized countries, they say parents participate in the paid labor market, which means that even young children spend a lot of time in child care. In Norway, more than 80% of one-year-old children attend child care centers and are brought up by a combination of parents and child care providers. Findings indicated good quality regarding the organization of meals and relational climate in about half of the centers. Sensitivity, language support, and facilitated exploration showed low quality across the different centers and are a cause for concern. Now, just for the record, it's this time in the second year when the left hemisphere is coming into a growth spurt, so this language is rather important because that's how Broca's and Wernicke's are going to move along when they come online at 14, 16, 18 months. They're going on to say, in this Norwegian study, none of the observed providers offered help to explore or expand the toddler's experiences during meals. Children at this age have very high exploratory needs. They're very social, they're very imaginative. They did not seem to register what the toddlers were interested in, e.g. other children. Overall, the observed providers were minimally involved in interactions and social interactions with the target toddlers beyond serving food during the meals. The almost complete lack of verbal exchanges between the care providers and the toddlers was also a striking observation in our study. They go on to cite Bjornsted and Oss found that providers in Norwegian childhood centers, childcare centers, often use few words when communicating with young children and only supply them with new words to a limited extent. This is especially worrisome as the second year of life is crucial when it comes to the development of the brain and central agency functions as logical thinking and language development, citing my own work and Judy's work. So the second factor would be these toxic endocrine disruptors. The slower development of males put them more at risk because of the extended window of exposure. Citing current increase in prevalence of developmental disabilities and autism, ADHD, and conduct disorders, Lanphier says the optimal strategy to prevent the development of brain-based disorders is to identify and restrict or ban the use of potential toxins before they're marketed or discharged into the environment. In addition to the national policy on early parental leave and improvement in child care, major contribution to our children's development would be governmental regulatory policy measures that protect our future citizens from early exposure to these environmental toxins. The research on the detrimental effects of relational trauma and toxins on early male and female fetal and infant brain need to shift from the later left brain language to early right brain social emotional functions. So I'll end with this. This is a study from the London School of Economics. What predicts a successful life? A life course model of well-being. And they conclude, the most important childhood predictor of adult satisfaction is the child's emotional health, followed by the child's conduct. The least powerful predictor is the child's intellectual development.
My name is Alice. I just wonder if ADHD or like autism is ADHD and autism caused by stress by environment, like from the biological, from the mother and the father, or is it some diseases that is natural? Or if you understand my question, does it come from the environment or does it come from the natural process, biological?
The HPA axis?
No, this autism and all those things that kids have that been diagnosed. Yeah, where does it come from?
Yeah. You know, more and more now we're starting to look at just what you've talked about, the immune system as being a key here, especially for positive bodily functions as well as for autoimmune diseases, etc. The autoimmune system is directly connected into the emotion processing system. So emotions can influence immune functions. As far as what the origins are of the early autoimmune systems, there's still a lot of work on that and still a lot of questions on that. Many of the autoimmune systems also have a gender effect, more females than males, etc., and very frequently they're associated with dysregulation, not of the testosterone system, but of the estrogen system, etc. But we're only in the infancy on looking at what those disorders are and how they can be treated, because most of the regular treatments are somewhat less than effective. That system also, though, is coming online very early in development, and I think that it is influenced by the attachment system. I think that it is socially influenced by the attachment system and by the corticosteroid stress regulating system.
Yeah, I'm John Downing again. I think I've more or less understood your explanations of the difference in vulnerability between boys and girls, males and female infants, but the question remains, why has it evolved that way? Have you any ideas or thoughts?
You know, the question, first of all, this idea that the male brain evolves more slowly than the female brain is found in all mammals. This is not just humans, okay? It's not just humans. My thought is that well, first of all, the reason why the right hemisphere would evolve before the left, because in terms of survival functions, the survival functions need to be there before language, et cetera, et cetera. But why nature has chosen to play it this way is a mystery for us to understand. And I think that the more that we understand about these situations, the evolutionary mechanism, the better, because we are starting to really heavily tinker with evolution, and it's in the end, it's going to really lead to a problematic situation.
It must be an evolutionary advantage for a female infant to be more robust.
All right. So the best guess for me would be the female carries the genome. She's the one who's going to send the genome down the line. So she needs to be preserved. That needs to be reserved. To be preserved first, so to speak. So I think it's for that reason. But that would be a guess. I mean, we all could make other guesses about why this would be. But what I want to say is we have to work. Evolution literally is the system, and then try to figure out why that would be, rather than, because Mother Nature doesn't make mistakes. Human beings make mistakes with Mother Nature.
But I've heard about survival of the fittest. That might be a good idea. Now there's a...
Hello, my name is Mari Vågevang. I'm a researcher at the Norwegian Institute of Public Health, and I'm heading a large project on daycare with a public health perspective. I have one comment and I have one question. And the comment is that when you look at research from Norway that represents large-scale studies, they don't really seem to find a negative effect on aggression in boys starting before the first year, or starting early, or even having long days. Actually, it seems to be a protective factor, especially for vulnerable boys, especially for language development. And also, if you look at the vulnerable children, then you find the effects, especially for vulnerable boys on aggression. But for most children, it doesn't seem to have, the Norwegian daycare system doesn't seem to be making children more aggressive. And that was just my comment, just to perhaps nuance the picture a bit. And my question has to do with learning and the school results, because in Norway we see that the gap between boys and girls is increasing in schools. And then I come back to cognition, but I'm wondering if you could use some of the same explanations to why more boys drop out of school and get poor grades, while girls seem to occupy all the student spots for the high grade study courses. So, yeah.
Right.
Could you use the same explanations for that?
Yes, I think you could, and because that gap, you're seeing that gap around the world actually, and it continues to spread out. You're seeing that also with with physical diseases, and males also this kind of gap. On the matter of the externalizing behaviors, my interest, first of all, is two issues. First of all, I think that the measures are important here. The measures, when you're looking specifically at cognitive measures, certain things may not be there. But when you get more and more into emotional function measures, social relational functions, which frequently I think are understudied, for example, just using language. Some years ago there was a situation whereby the major vector that was studied was whatever is happening early in development. It didn't really seem to affect the language functions. But again, when you're getting into more subtle social emotional information processing and emotional functions, it might show up there. These externalizing behaviors show up in other countries also. You may have something to do with Norway here, and because as you're well aware of, Norway is number 2 in UNICEF, etc. So you really may be pathfinders along these ways and closing that kind of gaps. But the other thing I want to say is there is more evidence these days that some of these effects do not show up immediately in a year or two, but they do show up later. In other words, if you really want to look at aggression and the influence of very early development on aggression, including fetal, because we know that very high fetal testosterone also has effects on externalizing behaviors, you may have to wait down the line. You may not see it at 2 years, 3 years, but in adolescence, when the brain is completely reorganizing, that's where it may show. That's exactly what was found in these endocrine disruptors. The effect of them was much more in adolescence on the reorganization. And that reorganization would not be the reorganization of the left hemisphere, it would be reorganization of the limbic system and its connection into the autonomic nervous system. So,
Excuse me, this will be the last one.
Hi, I'm Suzette. I'm a child psychologist. I was just curious when you told us that boys are more stressed in kindergartens and that they show more in an external way when they're stressed rather than the girls that show it more in an internal way. And my question is how can you know if the boys are more stressed, or can it be just that the girls are less visible because they are more internal, that they don't show it that clearly?
yeah.
Yeah?
2 points here. First of all, your use of the word kindergarten. All right. The studies that I showed were at birth and at 6 months and at 12 months. So birth and 6 months, and the differences there in male development, et cetera, was not. For me, the term kindergarten is much later in development. We don't think about kindergarten first year or even second year, et cetera. So I wasn't talking about that. Second of all, I want to pick up on your question. You're absolutely right. First of all, we are seeing an increase in female disorders also. And the truth of it is, let's now use in a classroom, let's use a first grade classroom. The little boy who was marching around the room, ADHD, he'll be picked up pretty fast and sent for care. The female who is sitting in the back, who has been experiencing PTSD, and as you know, PTSD is more common in females. Her cortisol may be going through the roof, but she's not activated and she's not forming unsocial behaviors, etc. So there's the other side of that. There's the spin of that also. Being the case, I want to leave though with you. This work that I've done is I'm a synthesizer, and so what I've done here is I've synthesized it. And so many of the measures that I'm looking at here are brain measures. They're not behavioral measures, as you would be using, etc. And we're looking at the structural changes within the brain here. So ultimately, the measures are a key here. But what I want to suggest is, and as I said, there were 10 other articles written on this kind of situation. And the real interest in the United States now is beyond externalizing disorders. It's a predisposition to violence. So now we're talking about rather serious sociopathic behaviors, et cetera, and the gender splits, with violence there, as we're seeing around the world with males also.